Hi everyone. As our chair said, yeah, it's a collaboration between different universities. We have been moving around for past few years because my supervisor changed his university. He was a postdoc at Massachusetts Institute of Technology. Professor Bhattacharya already knows him very well. So he's now an assistant professor at University of Groningen, and I am also under in his group, and I have worked with him in Nanyang and Smart Singapore as well. So our topic is Hydrogel-CNT Biomimetic Cilia for flow sensing applications. So just going through the contents, I will just briefly talk about biomimetics. I know most of you know about that. But most importantly, I will directly go to mechanosensory lateral line systems. And then I will talk about the superficial and canal neuromast found in fishes. And of course, talk about structural and functional significance of cupula, then the motivation of this work, and then directly go to our sensor, which involves the fabrication, then the morphological characterization, structural characterization, test, and then summarize it. So biomimetics, as we know, takes inspiration from nature to design efficient engineering products. Um, our project is inspired by hair cell mechanoreceptors. They are a class of highly efficient sensors found in many organisms, including fishes, insects, mammals. So they have been of immense interest to scientists and engineers because of their exceptional flow sensing capabilities. Substantial amount of work have already been done in past by Professor Chang Liu's group and others in the area of lateral line sensors. Uh, the lateral line sensors are a class of sensors which fishes use. They are placed directly on the skin of fish, and the individual unit of a lateral line system is called neuromast. Neuromast structure can be of two types depending on their structures. It can be superficial neuromast and canal neuromast. Now, blind cave fish, the first image is it's by the name, it's blind, and it navigates in underwater environment using these lateral line systems. So a neuromast consists of hair cells, which is connected to afferent nerve way to the brain, and we have the cupula dressing over that, which is like a hydrogel material, and it's supported by cupular fibrils, and the stereocilia bundles are deflected when the cupula gets deflected in a water flow, and it sends signals to the brain. So we directly go to the structural and functional significance of cupula. So cupula, of course, functions as scaffolding structures with the cupular fibrils to support the viscous hydrogel of the cupular structure. And by doing that, the ratio of hair cell is greatly increased, enhancing the sensitivity of the sensor because, of course, the drag force is scaled proportionately. And fibrils in the cupular structure also aid in coupling the hair cells, which are connected to the afferent nerve rootways to the brain directly. So the coupling is good, and the cupula of canal neuromast of certain fishes, which is shown on right, uh, have possess a very high degree of order in their structure. They are elongated structure in some fishes. Our work is inspired from the elongated canal neuromast structures found in some fishes. So, as I talked about the neuromast, there are two types of neuromast, canal and superficial. But the key difference between the superficial and canal neuromast lie in the shape of their cupula. If you see the superficial neuromast on the left, it's a schematic diagram as well of a sensor which we designed a few years back. The shape is called a prolate spheroid shape, whereas for canal neuromast, it's a hemispherical shaped sphere. Those two cupulas are very different, and the way they behave are very different. And also the material properties of both the cupulas are different. One is in the order of 8 to 10 pascals. In case of canal neuromass, it's like 10 kilopascals. And superficial neuromass acts like low pass filters, and canal neuromass act like high pass filters. So due to their structural differences, they respond differently to different frequencies. Now, superficial neuromasts are like low pass filters which react to low frequency stimuli, whereas the canal neuromasts behave like high pass filters and they behave to high frequency domain. Now, combining both of these type of sensors in the lateral line systems Fishes are able to distinguish like low frequency noises which are generated by swimming fishes from high frequency coming from some predatory animal or a prey. So the fish can distinguish between them quite clearly. And so the motivation of our work is investigating the features of the canal neuromast. So we designed a biomimetic cupula first, and we assessed its contribution to enhance sensing performances. So we developed and characterized an ultra-sensitive cell pod flow sensor using a combination of soft material processing and nanofabrication techniques. The sensor features an electrospun polyvinylidene fluoride nanofiber sensing membrane with a hyaluronic acid infused high aspect ratio vertically aligned carbon nanotube bundle, which mimics the biological cupula and the cupular fibrils. We have conducted some oscillatory tests on the sensor. I will be showing them as well. So bio-inspiration in these works come from two things: the cupula, which is mimicked by the HA hydrogel, it's a hydrogel material, and the cupular fibrils, the fiber-like structures inside the cupula, which is mimicked via the carbon nanotubes, vertically aligned carbon nanotubes. So we are motivated to biomimic the cupular structures, where our focus was not only to improve the sensitivity of existing sensors, but to devise a simpler biomimetic sensor with a cupula while using PVDF membrane as the sensing element. Some advantages of this mechanism of using CNT cupula-based sensor is, of course, high aspect ratio of the CNT bundle, then porous carbon nanotubes holding hydrogel between the nanostructures. They couple the cupula well to the PVDF sensing membrane lying beneath it. And of course, there is the high mechanical strength. Most importantly, we are simplifying the whole sensor design process. As we know, if we work in a clean room environment, fabricating those high aspect ratio structures, cylindrical structures, can be sometimes complicated. This is a very simple fabrication method I will be showing, and there we can achieve some sort of repeatability in that as well. So basics, we have just used a polyvinylidene fluoride electrospun nanofiber membrane for our work. We have used a rotating mandrel collector, and on the right is just a small video showing how it is electrospun. So when you apply voltage it goes on the collector, fibers get spun on the aluminum substrate. Yes, proper and 1 millimeter cavities, and then transfer our polyvinyl fluoride nanofibers on top of them and align the second layer of particular layer adhesive films. So we are making a suspended diaphragm of PVDF nanofibers, and on top of that, we transfer vertically aligned carbon nanotubes. If you see the figure G, you can see a vertically aligned carbon nanotube bundle standing on the suspended PVDF diaphragm and two gold electrodes coming out, which acts as the sensor output. And then in the last image, we dressed it with the hydrogel cupula, mimicking the canal neuromast found in fishes using drop casting method. So some ACM images of the sensor fabrication. First one is of course an optical image of the carbon nanotube bundles grown on silicon wafer. The second is an FE-ACM image. The third one, when we zoom inside it, we can see parallel alignment of carbon nanotubes inside the bundle. Image D on the left shows when you dropcast it with hydrogel, it swells up and the alignment of the carbon nanotubes get disturbed, and the carbon nanotubes spread equally in the hydrogel material, thus mimicking the cupular fibrils. And the image E shows you some ACM images of the nanofibers which we have used. They're nice and uniform. And the last one is, of course, a cross-section diagram of our sensor. So what happens with the hydrogel is the image on the left shows the carbon nanotube bundles without any encapsulation. When we encapsulate them with hydrogel, we have used HATI word, which is called Corgel. It's commercially available. And by varying the photoinitiator concentration, we can mimic the Young's modulus of the fish cupula, which is 10.4 kilopascal. We have achieved that, and it's shown that when by the action of capillary action, when it sips inside the carbon nanotube bundle, it swells up and it mimics the cupular fibril structures which you usually find in canal neuromast of fishes. So with and without, we see the difference. And after that, we test the sensor. There's the schematic diagram of the sensor placed in a tank, a small beaker, and we subject it to oscillatory flow. Now, oscillatory flows are very common in underwater environments, and fishes react to those stimuli. So to prove with and without cupula, first we test the sensor without any cupula dressing, just the naked bundle. And calibration has been done both in air, just the water was replaced with air. And we can see we did from 1 to 100 millimeter per second of oscillatory flow velocity, both in water and air. And D and E shows raw output from the sensor at 5 millimeters per second and 95 millimeters per second. By doing this, we were able to measure the threshold detection limit of 5 millimeters per second in both cases. Uh, so those two shows the results in water, and the experiments were conducted at 35 hertz vibrating frequency of the dipole. Now, quickly demonstrating how the sensor works. We subject it to some airflow. The bending is not that much. What happens when we have a cupular dressing? Uh, as you can clearly see that the deflection is highly increased, and so this leads to enhancement in sensing performance. So what we did, we took both of the sensors and set them 25 centimeter, 2.5 millimeters apart, 2.5 centimeters apart, and then vibrated them with a dipole underwater, to both underwater and air to see the increase in performance. As you can see, the sensing performance shown by the red line for naked and blue for the cupula dress bundle, there is significant improvement when you dress it with the cupula. So two reasons for this performance enhancement. One is the fluid structure interaction. Of course, the cupula has a very porous structure. So that leads to an enhanced drag force, which directly transfers to the movement of the PVDF diaphragm beneath. And also, it's obvious that the cross-sectional area of the cilia bundle is increased due to a bulge of cupular dressing. So these two factors leads to enhanced performance, both in air and water. In water, the performance enhancement was in the order of 8 times. In air, it was 2.6 times. And we did some more tests like response of the sensor to 3 pulses of airflow, then response of the sensors to tap of human hand. Then we walked parallel and walked away from the sensor to show. And in each case, we saw some significant output from the sensor. We also discharged some liquid droplets to see the performance of the sensor, and it could detect as low as 2 microliter discharge in water. So the sensitivity was around 6.5 millivolt per microliter discharge. So in summary, we just fabricated a simple method, used polyvinylidene fluoride fiber membrane as the sensing element. We had a threshold detection limit of 5 millimeters per second in the lower end. And of course, this is a very simple fabrication process, and we expect more people will be adapting to this type of techniques in future. And this research was supported by National Research Foundation, Prime Minister's Office, Singapore. Thank you.